Hello, everyone. Ciao, shalom, hola, and welcome. Thank you for joining the Foundation Fighting Blindness International webinar celebrating World Retina Day. Currently, all lines are muted and without video. Please be aware that the controls are at the bottom of the Zoom platform. This control bar may collapse when it is not in use. To prevent the controls from collapsing, you can use the following keyboard shortcuts to toggle between options. If you are using Windows commands, press the Alt key, and if you are using a Mac keyboard, press Command and backslash at the same time, and this will allow your controls to always remain visible and accessible. Today's presentation is being recorded and is available with closed captioning. To activate this closed captioning, please select the ellipsis or three dots located at the bottom right of the Zoom window, then select Captions, and then Show Captions. Throughout the call, you can ask questions via the Q&A and chat features, both of which are at the bottom of the Zoom window. You can also submit a question by sending an email to info@fightingblindness.org. I would now like to turn the call over to Laura Fietta, the International Division Chair for the Victory for Vision campaign, who will kick off this presentation. Laura?
Hello there. I hope everyone can hear me okay. Welcome everyone, wherever you are in the world. Depending where you are, good morning, good afternoon, or good evening. I am Laura Fietta. I'm in London in the UK, and I am the Foundation Fighting Blindness International Chair for the Victory for Vision campaign. And I'm so happy to be here with you today. It's fantastic that we have come together from all corners of the world to address retinal eye disease. This is incredible. We have people here today from every continent. We have over 45 countries represented. This is a truly international event and a fitting way to mark World Retina Day. Like many of you here, I'm sure, I'm very excited to learn about advances being made worldwide that move us towards treatments and cures for retinal disease. This international webinar provides us with the opportunity to hear the efforts of scientists around the world who are supported by the Foundation and the remarkable research that they are undertaking to combat blinding diseases. My 16-year-old daughter has an inherited retinal disease, and the FFB's international reach and focus has been a game changer in terms of accelerating research. We want to stop these diseases in their tracks, better still, reverse them. Since my daughter's diagnosis 10 years ago, the research landscape has advanced dramatically. The science has really taken off. The scientists know what to do. They have clear strategies for developing treatments and cures for retinal disease. The goal is treatments and cures for these retinal diseases. And they are determined
so sorry to interrupt,
to achieve this in a truly... Hello?
Sorry, would you mind just maybe turning off your camera?
Shall I? Hold on.
Yeah.
Just stop. Okay. Is it... Can you get better now?
Just a little bit better, I think. Yes.
Is it coming through okay?
Yes, I think so. Better now.
Okay, so shall I continue? Okay, so as I was saying, the FFB's singular goal is treatments and cures for these retinal diseases, and they are determined to achieve this in a truly global way because that is the best way and the quickest way. The FFB, they're dedicated to funding, coordinating and collaborating around the world, working with scientists, industry, charities and patients globally. I have found the Foundation Fighting Blindness to be a guiding light, a fountain of knowledge and resolutely international in their reach and purpose, in funding the science, in co-funding the science with organizations worldwide, in bringing scientists together, in bringing patients together, in scrutinizing the science, and alongside gene agnostic approaches, embracing the challenge that there are over 350 genes and counting, aiming to focus on each one with exceptional scientific rigor, dissecting the problems and working with the scientists to reach the most efficient solutions to these diseases. Also, in line with their international resolve, the FFB has joined forces with Chapel in York. This is an international philanthropy agency, and it enables donors outside the US to take advantage of their domestic tax benefits when making donations. That makes a huge difference to increasing the sums raised. And I have to mention the FFB's world leadership in diagnostics too, which is a vital component in the FFB's international response. And of course, a key part of this is the FFB's My Retina Tracker registry. It's completely free to join, and it's set up for patients to use. It's the global patient registry for people with retinal dystrophy. And years ago, the FFB helped my family join. Here today, we have people affected with retinal disease from all over the world, and we have scientists supported by the FFB tackling retinal disease around the world. It's a truly global community, and this is how to get things done. It's the time to come together more than ever. Working together, all of us around the world, is how we'll beat retinal eye disease in the quickest way possible. With shared knowledge, shared resources, shared science, and shared determination. We need to do this at a macro level. We need to pool resources for a coordinated global response in order to end retinal disease, and the Foundation Fighting Blindness is driving this worldwide effort. As patients, there's so much we can do to help keep the momentum going. We're looking forward in the next few months to sharing news about the development of the FFB's first international chapter, which will be in the UK, and we will announce how you can get involved and help raise funds for inherited retinal disease by participating in the FFB's do-it-yourself or DIY fundraising program called Raising Our Sights. There have been some extraordinary research breakthroughs recently. The science is here and ready, and we need to keep the funds coming in to finance this scientific new dawn. We're moving ever closer to treatments and cures across the board of inherited retinal disease and dry AMD. There are millions of people affected by these diseases worldwide, about 4.5 million with inherited retinal disease, and a staggering 130 million with dry AMD. If we all come together, all support the science, and all have our data ready for the science, we can find treatments and cures far quicker. And with our support, the FFB is here, pushing that science forwards, always pressing to go faster and not stopping until each of these retinal diseases is banished to history. So now I am delighted to introduce you to Dr. Amy Laster, who manages the Foundation Fighting Blindness Grants Program as the Senior Vice President of Science Strategy and Awards.
Thank you, Laura. Again, this is Amy Laster, and it is truly a pleasure to join you today from my desk in the United States. Today's webinar, it's an exciting opportunity for us to share a sampling of research that makes up our global research portfolio. The Research Awards program, it really lies at the heart of the Foundation's mission to support new therapies for inherited retinal diseases, and is matched with funding mechanisms that intentionally seek innovative science strategies and enable scientists and clinicians across the globe to address all retinal diseases and research areas towards future therapies for all affected individuals. Today, you will hear from 5 funded scientists whose research is or has been supported by one of the Foundation funding programs, from career development award programs to multi-investigator collaborative awards. So I know you're not here to listen to me talk. So with this, I want to introduce our first research presenter, Professor Alberto Auricchio. Professor Auricchio, over to you.
Thank you very much, Amy, and thank you for inviting me here and share with you the interim results of our project, which I'm gonna put on the screen in a second. I trust you can see the screen now. I'm sorry. I'll put the first slide. And this is the project is around finding a therapeutic approach for inherited retinal diseases.
Dr. Auricchio, we cannot yet see your slides.
Oh, you don't see the slides? That's weird because it says. Can you see them now?
No, not yet.
Try again. Can you see the screen now?
No, you might just need to hit present on your slides, and that might bring it better.
I am on the presentation mode on the slides. This was working a minute ago. You are screen sharing. You still don't see the screen, right?
That's right.
Mm-hmm. To me, it looks that it's sharing. So I'll try it again, but let me check if by doing just the window. Are you able to see the screen?
Yes.
Okay, great. So as I was saying, our project deals with finding a therapeutic approach for inherited retinal diseases, keeping in mind that these are one of the most frequent causes of blindness in the Western population. And the challenging aspect is that this group of diseases has a high genetic heterogeneity, which means that there are many genes that are responsible for the same disease. And indeed, the graph shown on the slide shows that there are over basically 300 genes that are singularly involved in a form of inherited retinal disease. And of course, this poses the challenge of how treating these diseases, because we all know that gene therapy holds great promise for the treatment of this condition. But if gene therapy occurs via gene-specific approach, we would need to develop 300 specific drugs, which is in part ongoing, but it's of course a challenging aspect. So I'm gonna discuss about a gene-independent therapeutic approach. So a therapeutic approach that can be applied to this group of diseases independently of the underlying mutation. And how to do that. So despite being caused by over 300 different by mutation in over 300 different genes, these groups of diseases has a common hallmark, which is of the degeneration of photoreceptors in the retina, which are shown in the cartoon here, are the rods and cones located in the outer part of the retina. So we can take advantage of this common mechanism underlying photoreceptor degeneration to design a one-fits-all therapeutic approach. And the approach that the project is actually exploiting it's based on microRNAs. So microRNAs are small non-coding RNA molecules that are expressed in the cells of our body. And they are non-coding, meaning that they are not meant to encode for proteins. They are meant to actually bind other RNA inside the cells and control their expression, so by downregulating them. And one of the important aspects of microRNAs is that their overexpression or downregulation is being considered as a therapy for retinal degeneration, as let's say, a one-fit-all therapy for retinal degeneration. There are already some examples of this in the literature. So the group of Dr. Banfi at Teleton Institute of Genetics and Medicine in Italy and other colleagues at TGEM in Italy have shown that if we use viral vectors like those for gene therapy to overexpress or to block microRNA, we can have benefit in some models of inherited retinal degeneration. So based on these premises, the project that the Foundation Fighting Blindness is sponsoring, it's actually a microRNA-based therapy for inherited retinal diseases called also RETMIR. And this is a program project award, which means that involves 3 different groups. They are all European groups. And you will see in the slide the one group, it's actually the group of Dr. Banfi at TGEM, who's well known for his research on microRNA and studies of microRNA in the retina, both in terms of their role in the physiology of the retina as well as in diseases of the retina. And so he's the microRNA expert of this consortium. And there are 2 additional groups, my group at TGEM and the group of Dr. Dalcara at Institut de la Vision in France, who are experts of retinal gene therapy. So by combining our different expertise, we want to tackle this ambitious objective, which is shown in the next slide, which is a comprehensive screening of all the human microRNAs to identify 1 or more that have the strongest neuroprotective activity in the retina of mice with retinal degeneration. So to do that, the slides will show on the left a cartoon where we are basically cloning all the 500 putative human microRNA, as well as 300 different sequences that can block them into AAV vectors. So we are basically developing a library of AAV vectors that express at the same time multiple microRNAs or sequences that can block them, so that we are able to either express, overexpress microRNA, or we can downregulate and block them. So we take this large number, this large series of constructs of vectors, and we basically inject them directly in vivo in the retina, subretinally, in a mouse model of retinitis pigmentosa. And we look at those that have a protective effect in the mouse model of RP. So we do the screening directly in vivo in the retina of mice. And the way we are looking at the protective effect is a rather, let's say, conventional way, which is to look at either or at both protection in terms of electrical activity of the retina, functional rescue, there is a graph representing ERGs, or looking at the protection on the structure of the retina, so preservation of the photoreceptor layer in the retina of mice. So the slide that I'm showing now shows a graph, which is an example of a group of experiments where in the graph you will see multiple bars. And these bars represent the electrical activity of the mice after the injection of several of these different constructs. And you see all gray bars that are affected animals receiving potentially therapeutic constructs. And on the right, you will see a white bar, which instead represent the electrical activity of untreated mice as a negative reference. And if you look at this crowded graph, you'll appreciate that there is one bar that is higher than the other. And this is one construct that consistently has shown significant improvement of the electrical activity in the retina of mice, because of course, the higher is the bar, the higher is the functional activity of the retina, and so the higher is the protection provided by the treatment, by the subretinal administration of the construct. So I want to conclude by saying where we got with this in these first 2 years. So this is a 5-year project, and we have completed the 2 years of activity. So in these first 2 years, we have designed and generated the AV vector libraries, which has taken about a year now. And then we have started to screen this set of constructs in vivo in the retina of a mouse model of retinitis pigmentosa. And we are in the middle of this screening. So although we are in the middle, so this has not been completed yet, we have already identified one vector that protects better than the others the retina of the mouse model of retinitis pigmentosa. And while we are finishing the in vitro screening, we are already characterizing the protective, the vector in terms of selecting out the protective microRNA and its mechanism of action. And with this, I finished my presentation. I want to thank, of course, I'm speaking on behalf of 2 other groups, as I mentioned before, the group of Dr. Banfi and the group of Dr. Dalkar in France and their collaborators. And I am glad to introduce the next speaker, who is Fernanda Abalem from the University of São Paulo and University of Michigan. Thank you very much, and thank you to the Foundation Fighting Blindness for supporting us.
Thank you so much, Dr. Auricchio. I'm Fernanda Abalem, and I'm truly honored to stand before you today. Looks like we're over 400 people. This is an important event organized by the Foundation Fighting Blindness, an organization dedicated to making a profound effect in the lives of people with inherited retinal degenerations. Well, as we gather here today, I'd like to share a bit more of my personal journey in the field of inherited retinal dystrophies and the global impact it has had on me. My career has been an incredible odyssey that has taken me from the University of Michigan to the academic environment of the University of São Paulo, and later also in my own private practice. Well, I want to share with you why I chose to dedicate my life to the studies of the IRDs. My decision was, in fact, deeply influenced by the historical context and the urgent need for change. In the not so distant past, the landscape of IRDs was vastly different from what it is today. These conditions, which are often referred as orphan disease, rare disease, were sadly neglected by much of the medical community, and there were no available therapies, and research in this area was sparse at best. And patients in the long run affected by IRD were left feeling forgotten and under-cared for. So thank you to the FFB. I started my journey at the University of Michigan, where I received comprehensive training in the clinical and genetic aspects of IRDs. During this very important phase of my career, I had the privilege of learning from some of the foremost experts in the field. I was mentored by Dr. Jayasundara and Dr. Hecken Lively. I learned a lot with Ms. Brahan and Ms. Schlegel, a lot with Dr. Nahid Khan, to whom I'm very deeply grateful. And I immersed myself in the world of diagnostics, therapy options, and the profound impact of the diseases on individuals and their own family. Later on, as I ventured into the University of São Paulo, I was met with a different set of challenges and opportunities as well. My mission was to establish a similar model of care, one that would combine the cutting-edge knowledge that I acquired with the unique cultural and healthcare landscapes of Brazil and South America. One of our main missions, either in São Paulo or in Michigan, is to allow our patients to feel independent. The ability to read, whether it's a book or a street sign, becomes a difficult task. The simple joy of recognizing a friend's face or being recognized can be elusive as well. So independence to move around safely without assistance may reduce, even as navigating a familiar environment can become a hazardous task. Personal care routines such as dressing and cooking or maintaining personal hygiene can turn into a formidable challenge. So every step, every motion needs to be approached with caution and capability, constantly adjust to a world that seems designed to only those who actually have sight. So we focus a lot of as well in physical therapy, occupational therapy, low vision devices, and also we also take care a little. We take a deep and an important care with the emotional struggles that patients with IRDs might face. The emotional struggles are as real and as profound as the physical ones. Most of you might know that individuals with IRDs often grapple with a sense of loss, grief, isolation, the realization that their vision will continue to deteriorate, and the fear of what lies ahead can cast a long shadow over their lives. Here we have a painting of Salvador Dali with the Perseverance of Memory, which shows us much of what time means and how time deteriorates. And time, it's actually not a good friend of a patient with IRDs. And relationships can be strained as they grapple as well with feelings of dependency and vulnerabilities. But this is not just a personal impression, it's science. Our group from Michigan, led by Dr. Jaya Sundara, has made a lot of work addressing the impact of the psychological distress on vision of patients with IRDs and how they affect their daily lives. So we have had a questionnaire that addresses the psychosocial outcomes for IRDs. We have had research how vision-related anxiety and the perception of vision as well, either if patients with retinitis pigmentosa, with selected groups of patients with EYS, and the list goes on. And here, it's important to know that you are not alone, and science has made a tremendous stride in the field of gene therapy and cell therapies, offering hope for a brighter future for patients affected with these conditions. And as we celebrate the scientific achievements today, let's also remember that addressing the psychological and emotional aspects of this disease is just as crucial. So it's a key to see better. You have to feel better as well. Thank you, Foundation Fighting Blindness, for its unwavering dedication to this cause and for organizing this event. Let's also use this day as a reminder of the importance of our collective effort, our shared goals and dreams, and commitment to make a difference in the lives of you all which suffer with IRDs and low vision on a daily basis. Thank you. Now I will introduce Dr. Manuel Irimia, which is a bright researcher from Spain.
Thank you. Thank you, Fernanda, for the presentation, for the introduction as well. And of course, the Foundation Fighting Blindness for the kind invitation, and of course, for the support. Well, what I will tell you about today might sound a little bit cryptic for most of you, but hopefully towards the end of it, you will see it's an interesting research topic. And what I will tell you about is how very small elements of some genes that we call microexons are actually important to specialize the proteins that play crucial roles in photoreceptors. And therefore, these microexons might be missing factors in understanding retina disease. So basically, the core of this project and many other things that we do in the lab is a process the cells use that is called alternative splicing. And this process is very interesting because it really helps to diversify the flow of information that goes from DNA to RNA and proteins in the cells. And so the way this works, here I'm showing you an example gene of our genome. And I guess you know, but you have to keep in mind that the information, the genetic information in our genes is not encoded as a single unit, but is broken into pieces that we call the exons that are interrupted by non-coding sequences that are called introns, and depicted here as lines. And when there's a transcription of the gene happening, then we need to remove these introns and put these exons together to form the mature RNA. And this is then what will give rise to the protein. So you can think of these a little bit as our genes being a sentence in which the words that form this sentence are these exons. So we can directly trace a given exon, so a given word, to then the mRNA sequence and the protein. Okay, so if you think about it, then the beauty of it is that then the cell can play around with these words, with these exons. So for example, it can omit one of these sentences, one of these words in the mRNA. So now the sequence will not be present. And so we have a protein that is slightly different. So it will have a different meaning, a different function in this case. So this is the process of alternative splicing. And as I said, it's very interesting because we know tons and tons of cases in which these different isoforms, these protein isoforms, can have different functions, cellular locations, etc. Now, another thing that makes it very interesting is that this process can be regulated very finely in a cell type specific manner. So we can have, for example, one protein isoform that is only present in the neurons, whereas the other cells have the other protein isoform. So that allows for a very fine specialization of the proteomes, which is the total of the different proteins of a given cell type in a very cell type specific manner. So this is the general process that we work on, but in our lab, we're particularly interested in a given type of alternative splicing and a given type of exons that we call microexons. And as you can imagine by the name, these are very tiny exons. We define them as encoding as little as 1 to 9 amino acids in a protein. And you have to think the normal exons, or the normal word, would be around 50 amino acids. So they're very tiny, but despite being tiny, we know they're very functionally interesting because they lie in very specific regions of the protein and the structures of the protein. And what they do is modify the way proteins interact with each other. And what makes them even more interesting is that they appear mainly in the neurons. So now, if you put it all together, you can think that there's a few little tiny microexons that change the proteins a little bit so that they do their function in a very specialized manner in the neurons, whereas the other cell types have the regular isoform with all these microexons. So this is something we really work a lot on, microexons. And of course, at some point, we thought it was a great idea to study them in retina and in retinal diseases. So why would this be important? Well, obviously, for most of you, it's an obvious question, but even from the splicing perspective, it's quite interesting. Because we know, of course, that many of the retinal diseases, most of the retinal diseases are characterized by the degeneration of the photoreceptors. And these cell types are really fascinating because they're extremely complex, extremely elaborate. And I'm just depicting here a very simplified scheme of a code and a rod. And you can see their unique structure compared to any other cell type, especially this outer segment, which is a very modified cilium. And therefore, to achieve this function that it has and the structure, it requires a very specialized proteome. And as I said, specializing proteomes is really a key asset of this process of alternative splicing. There are other ways, of course, but alternative splicing is very good at this. So that's the first reason. But also, we know from work of many different people that somehow this cell type, the photoreceptors, are very sensitive to splicing mutations. So mutations in splicing factors, splicing regulators that don't have any effect in other cell types really cause phenotypic defects in the photoreceptors and are cause of some of major these retinal diseases. Then another reason is that there's even some of these isoforms that are specific of the retina that are known to be mutated and cause disease. And finally, a bit of a different reason, more of a practical one, is that the retina is very amenable to targeting to treatments with antisense oligonucleotides. And this is a new type of molecule that is really, really important in therapeutics right now, and is perfectly suited to modulate splicing. So we can modulate alternative splicing and perhaps help correcting diseases. Now, with this in mind, then we decided to study how microexons look in the retina, and to our surprise, or let's say happiness in a way, we found the retina microexons are the retina has the highest number of tissue-enriched microexons. So this is telling us that the retina, as we hypothesize, have a strong specialization, uses these microexons more than the brain, and of course more than other types like muscle and heart. But this is in human, and this goes even to other organisms that we look at. So we look in mouse, in chicken, and zebrafish, and in all of them, the retina has the highest number of tissue-enriched microexons. So then what we thought was to look for those that are really peculiar or particular of the retina, and this is what we call retmix, so retina microexons. And we identify around 70 of them in humans. We divide them in two categories. One are really microexons that are only present in the photoreceptors, in the mRNAs of the retina, and not in other cell types and tissues. And others are very highly present in retina, and much less in the other tissues. And whatever the case, they are really especially regulated in retina, so we thought they could play important functions. So the first thing we did was to look at the type of genes that contain these retmix, and we see very nicely that they are usually genes related to ciliary assembly, vesicle-mediated transport, etc. And these are, as I say, key functions to establish this outer segment, the specialized cilia of the photoreceptor, and vesicle transfer, for example, is very important for the function of it. And not only that, but we also see very strong statistical or a significant statistical enrichment for these genes being associated with known retinal disease. So all in all, this is telling us that these retmix are in genes that are important for photoreceptor function. So then, of course, we wanted to see how we could see the impact of the functional impact and relevance of these exons. So the first thing we needed to do was to identify who is regulating it. What's the machinery in the cell that makes sure that these microexons are present in the retina? And for that, I'll spare you the details here, but we look at our favorite candidates, because there's a family of splicing factors called SSRN3 and 4. You don't really need to care about the name. But we knew that these regulate neural microexons. So we knew they could be regulating also these retina-specific microexons. So we did an assay that I'm showing here, in which you take this gene, SRN3, SRN4, and also Musashi1, which is a gene that is known to be important for photoreceptor splicing, and we put them in cells that are not photoreceptors, like HeLa cells normally used in the lab, and check if this could make the microexons be present. And in fact, we saw this for SSRN3 and 4. Basically, just by putting them in any cell type, we can force the inclusion of these microexons that otherwise would be only in the retina. So now that we have these two factors identified, and we look how they behave during development, and we can see that one of them, SSRN3, of the two paralogs of the family, is the one that stays up in mature photoreceptors. So we decided to focus on it. And then we switched to zebrafish, which is one of the model organisms that we have in the lab. And here we use CRISPR-Cas9 to remove or mutate a functional domain in this SRN3 gene and create it two mutant lines. And then we decided to take a look at it. And the first thing that we need to do in these cases is to make sure that if we think this effects is important for microexons, retmix, then we was to check on the transcriptome that this was the case. This graph is a bit complicated, but I'm just showing you how misregulated these exons are. And you can see the more to the left they are, the more misregulated they are. And you see retmix are the most misregulated. And also, what we saw when we analyzed the transcriptomes is that there was a strong, very strong enrichment for downregulation of genes that were important for phototransduction, perception, eye development, etc. This is telling us strongly that our mutant had issues with the eye development and probably vision. So we then, of course, started to investigate this more thoroughly. And obviously, as we expected, our mutants were visually impaired. We did this together with Stefan Neuha's lab. And well, you can see, so in this graph, basically, I'm showing you how the SSRN3 mutant responds less to light at different intensities. Now, how is this? Well, then, since they don't see well, we hypothesized there would be developmental issues and structural malformations. And I'm showing you here histological analysis of the retina. This is how a wild type looks with the different layers. And we see that only the layer that contains the photoreceptors is affected. It's reduced. And then when we study this in more detail, for example, using an antibody that stains rhodopsin, well, in the wild type, we see the outer segment very well labeled. In our mutant, we see that this outer segment is much shortened and there's mislocalization of rhodopsin. And this is at 5 days of development of the larvae, so very tiny fish larvae. At 10 days, just a little later, there's almost no photoreceptors, so they really start degenerating, and this goes together with apoptosis, so cell death. Now, all that is a summary of the work that we did. Of course, characterize this in more detail, but I hope it's enough to convince you that these microexons could be relevant for retina development, function, and obviously diseases, but they had not been studied at all before. So what hopefully we'll be able to investigate with the support of Foundation Fighting Blindness, it's these as well as other splicing mutations. So we haven't started the grant yet. It starts actually in a week. But the goal is to identify new splicing mutations in patients with retinal degenerative diseases, and of course, focusing especially on red mix and other regulated exons. Of course, we will identify any possible splicing mutation, but really our focus is on this. And the strategy is in a way straightforward, and we will start by sequencing the genomes of 87 patients provided by 2 institutions, the IMO in Barcelona and the University of Campania in Italy. And the key thing is that these patients, we know they have one identified pathogenic allele, and therefore we suspect that there's a mutation in the other allele that has been missed by the exon sequencing, and we think they might be splicing related. Not all cases, but hopefully so. And just to finish, then we will simply identify splicing mutations, validate this using the standard mimicking assays, and then validate this for some selected cases using organoids of retina derived from pluripotent cells of the patients. And then, very key, we'll try to design very effective antisense oligonucleotides to correct these splicing defects. And then we'll attempt to correct this in retina organoids. And hopefully we'll be able to have some candidates that we can go a step further in application. And with this, I just want to finish by acknowledging the people that did the work. And basically, this is Ludo, a great PhD student. That I'm very lucky that she will stay to develop the project with Foundation Fighting Blindness. And she has done her PhD together in my lab and the lab of Luis Serrano. So many people in the lab help them. And then our collaborators. And I also want to thank Sandro, who was already mentioned in this meeting. So hopefully he gets a lot of counts of people acknowledging him. So thank you very much. And with this, I'll stop sharing my screen. And I will introduce the next speakers, which are Dr. Alison Hardcastle and Dr. Mike Cheetham from the University College of London. And they have a program project award. So all yours.
Thank you, Manuel. Okay, let me share my screen, hopefully successfully. Let's do this. Share. Okay. Can you see my screen?
Yes.
Yes, we can.
I will do the
slide. Not yet.
Okay. So thank you very much. So today I'm going to describe our program project award from the Foundation. This is a truly collaborative award that supports our collaborative research. And our research is comprised of 3 research teams. One of the research teams is led by Suzanne Roosing at Radboud in the Netherlands. The other research team is led by me here at University College London. And the third team is run by Mike Cheetham, also at University College London. So our research is focused on one of our new discoveries. We've discovered a new genetic cause of retinitis pigmentosa. I'm sure you're aware retinitis pigmentosa is first characterized by night blindness and tunnel vision, but is progressive. And the image I'm showing here is the human genome with a big arrow pointing to chromosome 17. And this is where we've discovered a new genetic cause for RP. Now, what we've done in this collaboration is we've looked in families in the Netherlands and families in the UK and found this cause of disease. And in fact, we've also looked at families from South Africa and families from Canada. This is a dominant form of retinitis pigmentosa, which means that an individual with RP17 has a 50% chance of their children inheriting this condition. And actually what surprised us when we made this discovery is within these families there are hundreds of affected individuals with this form of RP, and we call this form of RP RP17 because we found that it's on chromosome 17. Okay, so why is this such a new and unusual form of retinitis pigmentosa? Well, normally when we do our genetic studies, we find genetic changes in a gene, and that genetic change alters the DNA sequence and then alters the function of that gene in the retina. However, what we found in RP17 is structural abnormalities of the actual chromosome. So chromosome 17 is altered, not a gene change. So this is very unusual and very new and rather complicated to understand. What we think this is doing, well, certainly genetically what it's doing is it's creating more copies of multiple genes on chromosome 17 in our patients. And we call these structural variants. Now, normally on chromosome 17, you will have a gene, let's call it gene 1, and that gene will be close to an on switch. That on switch is really important because it makes sure that gene 1 is expressed in the retina, and that gene is very essential for a healthy retina. So that's what we see in a normal chromosome 17. What we're seeing in these structural variants with in our patients with RP17 is other genes are now coming in to close proximity in the same region on chromosome 17 as the on switch. Now, the on switch is important to switch genes on in the retina, but these other genes are not required to be in the retina. So we've developed this hypothesis that these structural variants are actually being switched on by mistake in the retina. And our hypothesis is that this could then perhaps be toxic because it should not be there. So this is our working hypothesis. And what we want to do, our major questions are how many families have this form of RP worldwide? We need to understand the prevalence of this condition. How do these rearrangements of chromosome 17 cause retinal disease? It's not that we've got a mutation in a gene. And then, of course, the ultimate is, can we develop an effective therapy or treatment to prevent or slow down progression of retinitis pigmentosa for this condition? And for this, we're using lots of cutting edge technologies and experimental tools across our 3 research teams. So team 1 is led by Suzanne Reusing in the Netherlands, and her real focus is on the genetics. So we've got the families that we already identified with structural variants, but we have, of course, many unsolved cases across the world with dominant retinitis pigmentosa who don't know what the cause of their condition is. So we're doing lots of very detailed genetic analysis in these families to identify if any of these families are also RP17. And actually, we're finding many different structural variants. So not all families have the same change in the structure of chromosome 17. Some might have 2 or 3 genes entering this region near the on switch. Some might have 3 or 4, but they're actually all in the same region. So what we're tasked with here is, what do all these RP17 families have in common on this region and on chromosome 17? And it looks like there might be one gene that keeps popping in next to this on switch that might be the culprit. And this is our working hypothesis. And actually, they've made huge progress, and we can now see some more countries that we've represented, including the United States and Germany. We found RP17 families in these countries as well, with different structural variants. Because we know where to look and we know what we're looking for, we can really rapidly now find new families with this condition. And the team in the Netherlands are looking at cohorts of unsolved cases and families in Australia, Japan, and other European families. But I said, you know, as I said previously, we've identified hundreds and hundreds of affected individuals with this condition. So again, what's surprising is this is actually quite a common or major cause of dominant retinitis pigmentosa that was previously unidentified and unrecognized. The second team, which I lead, is really focused on understanding what is causing the retinal degeneration as a result of these chromosome 17 alterations. Now, to do this, we're creating a retina in a dish as a model to identify the culprit, which we think is gene 2. And this is an extraordinary thing that we can now do in the laboratory. So we can take a skin sample from a patient with RP17, grow it in a dish in a laboratory. We then can reprogram those skin cells and make them into pluripotent stem cells. This then enables us to take those stem cells and push them into making a little mini retina in a dish, which is just an extraordinary thing to see in the laboratory and really enables us to do lots of experiments that we couldn't do before. And we're using this, I've put a picture on this slide that shows you how beautifully these little mini retinas recapitulate what's actually the structure of the retina in a human eye. But we have this in a dish so we can do experiments on these little retinas from the patients to work out what's going wrong in this particular retinal model. And I'm pleased to say that the experiments we've done so far supports our hypothesis using these retinal organoids, and we can see that gene 2 that should not be switched on in the retina is actually switched on as a result of these structural variants. So our working hypothesis is looking good. We still have a lot of work to do, and we need to determine if gene 2 is really toxic and a bad thing to be in the retina. So I'm just going to hand over briefly to my colleague who runs the third team, Mike Cheetham, to talk about his aspect of the programme.
Hello, thank you, Alison. Hello, I'm Dr. Mike Cheetham. I'm also in London, and I'm the lead of Team 3. Now, our aim here is mainly to develop and test a therapy. Now, Alison's told you about the retina in a dish model, which is a brilliant new advance in understanding how genetic changes lead to human disease. And although we can do amazing things with those now, they still have limitations and they're not quite the same as the retina in an animal. And so to complement those studies and to complement the work that Team 1 are doing, we're going to make an animal model. So what's shown on the slide here is our strategy for making that. So we're going to take a control mouse and we're going to use molecular scissors to do gene editing and create a mouse with the same structural variant that we find in this case, it will be the family from the Netherlands, because that at the moment is the simplest duplication, the simplest structural variant we've identified. And so we're trying to recreate the change that's found in the Dutch family. Once this is currently underway, so we've already done the gene editing, and we have some mouse pups, and they're going to be screened in the next few weeks. Hopefully, we will have a mouse that has RP. To test that, we will take the mice and examine their retina and their vision, and we will also be able to compare those retina with the human retina in a dish models, which would let us see how well the human and mouse recapitulate each other. Now, when we have these mice, we can also then use them to test different therapies and use those same therapies on the retina in a dish model of the human retina. And we have several different approaches to doing this. we can reapply our molecular scissors, we can apply viruses, or we can apply the small antisense oligonucleotides that Manuel introduced in the last talk. And these are several different approaches we can attempt to use, and we can test them both in the retina in a dish and in the mice. And then we can ask the question of if they actually have any improvement in their vision or how slowly their retinas degenerate. Back to you, Alison.
Okay, thank you, Mike. And just for the last slide, I really wanted to emphasize the importance of this program project award from the foundation. We have 3 teams here who are highly collaborative with different but interconnected goals that we hope we can make a huge difference. It's a privilege to work with such an excellent collaborative team, and it's a privilege to work on such an important research, especially, you know, for the RP17 families. Patients are at the heart of our collaborative research efforts. Now, we have nicknamed ourselves the Dream Team because we're enjoying collaborating so much. It's tremendous. And I just hope we can live up to that nickname. And on that note, I'll finish. Mike, if you want to hand over.
Thank you, Alison. And thank you to the organisers, and thank you to FFB for the support. It's a great pleasure to introduce the next speaker, Dr. Eyal Banin, who's a clinician scientist and will tell you about his exciting work.
Thank you very, very much. It really is an honor and a pleasure to be here. And I thank all the people who joined this webinar. And what I'll try to do in the next 12 minutes is, after a short introduction of the diseases that have been talking about, that have turned into the testing ground for many novel therapeutics in the recent years, I'll describe our major long-term effort to try to characterize phenotype and genotype in the Israeli and Palestinian populations. An effort that, thanks to FFB support of a nationwide consortium, is enabling us to move towards our goal of having actually a nationwide database of patients in terms of their phenotype and especially their genotype, as this may serve both for prevention and for application of treatment. And I will give a few examples in which this indeed occurred. And I will finalize by finding within this work an effort of the gene that causes the most cases of autosomal recessive RP in our population, which is rare in other populations, which is called FAM161A, and our efforts to develop treatment for this specific gene. It's not moving. Just a second. Sorry. Oh, there we go. Okay, so as we know, IRDs are very clinically and genetically heterogeneous. There are many phenotypes, and over the last decade or so, this group of diseases is undergoing a revolution that started with the progress in molecular genetics that allows to identify the causes of disease, and this led to development of various gene-based therapies, but this is not the only types of therapies that are now entering into the arena of these diseases. We also have cell-based therapies, neurotrophic factors, and also visual prosthesis, attaching hardware, if you will, to the central nervous system. And the reason that the eye became such an ideal target, an ideal testing ground, is that it's really a wonderful tissue for testing these different new therapies. It's a small confined organ. It's accessible by surgery and injection. By delivering locally, we need to introduce only a small amount of medicine or cells or viral vectors, such that systemic toxicity or risk is lessened. We have very good ways to follow visual function and also retinal structure and function non-invasively to see the effects of the treatment. And last but not least, if complications do occur, they can usually be contained, sadly, perhaps with the price of an eye, but not with the price of a life. So many new therapies are making their first steps in our organ and in this group of diseases. Our journey to try to characterize phenotype and genotype in our population started over 20 years ago. When you see in the picture, Professor Dror Sharon, who's my friend and collaborator over these years, he's a molecular geneticist. And we started already towards the year 2000 in recruiting families with retinal degenerations and trying to characterize the disease and the genes. And you see here the graph of accumulation of families. And today we have over 2,300 families, over 4,000 patients in the database. But the big push came when the FFB in 2014, thanks to the efforts of Ido Perlman from the Technion, who coordinated this effort, they gave us support for a consortium that includes the main IRD centers in Israel to pull together the efforts and try to reach what I described earlier as a nationwide effort to characterize the disease in as many of the patients as possible. And today we have over 3,000 families, over 5,000 patients, and based on prevalence analysis, we think that we probably recruited over 50% of the families with IRD in our population, and we are striving to continue this. And the idea behind this is, first of all, to identify the phenotypes and the common genes in our populations, seek novel genes and mutations. We always notify the patients and the families of the genes and mutations that were found in order for them to be able to prevent further birth of children affected by the disease in the family. And we're also using the findings to explore and apply a novel treatment. So this is a list of genes, novel genes that were found to cause different forms of inherited retinal degenerations. Some of them were found at our centers within the consortium, some in collaboration with good friends around the world. And this is two pie charts that I'm showing here. On the left is the phenotype in the Israeli Consortium of Genetic Eye Diseases. And like other populations, the main phenotype we see is RP, followed by cone rod dystrophy, Stargardt disease, and Usher. And on the right, you see the main genes causing disease in our population. And like other populations, ABCA4 is a major cause, followed by USH2A, but then we can see FAM161A as the main cause for recessive RP. And we also see a high prevalence of a disease rare elsewhere, achromatopsia caused by CNGA3. And this has focused our efforts on these genes. So what has the genetic diagnosis through the consortium assisted in? So first of all, many of our families are already utilizing PGD, pregestational diagnosis, to assure that they give birth to non-affected and healthy children. In addition, we now have a very significant percentage of the Israeli patients that are genetically diagnosed. About 65% have their genetic diagnosis, and thus they're candidates for treatments that are already out there or that are becoming available. And some examples of that are the fact that we identified a founder mutation in the RP65 gene already in 2004, and these patients were amenable for treatment, and we had a small phase 1 gene augmentation clinical trial already in 2010. But of course, when Luxturna became available, we were able to reach these patients, and those that were eligible in terms of residual photoreceptors could receive the treatment. And in addition, now we are running different trials and treating patients that were identified through this phenotype genotype effort with mutations in CNGA3 and CNGB3 causing achromatopsia, as well as RPGR causing X-linked RP. I'd like to devote the last few minutes of my presentation to describe our journey to try and develop treatment for the main cause for recessive RP in our population, a gene called FAM161A. And it started in 2010 when we identified this gene as a cause for RP through characterizing the disease in a large cohort of patients, forming a knockout mouse model and characterizing it, and then attempting treatment in this model. So Dror Sharon was able in 2010 to identify that mutations in FAM161A cause RP, and this was in parallel to Carlo Rivolta from Switzerland, finding this also in a number of European families, and this was published back to back. And at the time that the gene was diagnosed, the role of the protein that it encodes was not yet known, and we and others about a year or two later found that it encodes for a ciliary protein that's mainly localized to the basal body of the connecting cilium of photoreceptors and important for photoreceptor function, both rods and cones. And over the years, more and more families and more individuals that manifest RP due to this gene were identified. We see on the slide some of the families, and on the right in the pie charts, we see that many of these patients are of North African Jewish origin. It turns out that there are a number of founder mutations in this population, and the prevalence there is very, very high. In terms of the phenotype, it's similar to other forms of RP with the age of onset between the first to third decades. The initial symptom is night blindness, followed by visual field loss. Interestingly, most of the patients have moderate to high myopia with a mean of over -6 diopters in a cohort of over 100 patients. I'm showing graphs here showing that the visual acuity over as a function of age in the different patients. I won't go into the different mutations, but what we can see is that in this cohort of 100 patients, many patients will maintain a visual acuity over 20/50 up to the 5th decade of life, suggesting a window for treatment. And the OCT supports this because many of the patients maintain a small island of intact outer nuclear layer or photoreceptor layer and also ellipsoid zone. And this is, again, a window for treatment and opportunity because this is retained in many patients up to the 5th decade of life. So we see now on the slide a sort of a rough roadmap for an attempt to develop gene augmentation therapy for a genetic disease. So now we have the gene in hand. We want to target FAM161A. We need to develop an animal model. And indeed, we produced such a knockout model. And this model manifests disease. We characterize the disease course. This is showing that the visual acuity of these mice as measured by the optokinetic response deteriorates over the first 8 months of their life. And by 8 months, they cannot follow the grading surrounding them anymore. And this correlates with what happens to the ERG, the electrical responses of the retina that deteriorate gradually over the course of 8 months and become almost non-detectable at 8 months of age. In vivo OCT imaging that can show us the structure of the retina shows gradual thinning of the outer nuclear layer, the layer of photoreceptors over time. And perhaps the most convincing evidence is the quantification of the thinning of the retina, and particularly of the ONL or photoreceptor layer. And you can see the graphs as well as the histology images showing that we were gradually losing photoreceptors until at 8 months there are only sparse nuclei remaining. So in many ways, this resembles a human disease. It's slower than other mouse models like the RD1 and RD10 models, and also gives a window for treatment as the progress of disease is not very, very aggressive. So the next step towards gene augmentation is to develop the viral vector that will deliver the normal gene to the photoreceptors. And here we have wonderful collaborators in Switzerland from Lausanne, Dr. Ivan Elsinievich and Dr. Corinne Kostik, who manufactured for us a couple of AAV vectors that carry the normal FAM161A gene under the control of the AGRK1 promoter and IRBP enhancer. And I won't go into the story of isoforms. We heard a little bit in the previous talks that this has a big role in the retina. But to cut the long story short, we found that the long isoform of FAM161A is more important. And what we did is we injected the vector under the retina in the knockout mice to see if this can provide a rescue effect. And we followed the status of visual function and retinal structure after this treatment. And the way we injected is that we either injected only one side of the retina, the nasal side with the vector, or we injected both sides. So in the cases of a unilateral injection to one side of the retina, we could always compare to the other side of the retina in the same eye. So here you can see on the top row, fundus autofluorescence images from a knockout mouse, and we can see that we have FAF changes accumulating over time as the disease progresses. And then in the middle row, with the green arrows, you see the treated nasal retina in which the changes are very slow to occur, as compared to the temporal retina with the red arrows, which was not treated, where we see the FAF changings accumulating. And below is a double injection eye in which both sides of the retina were treated, and we see that the changes in FAF are much slower to proceed. The same for looking at the ONL layer, the thickness via in vivo OCT serially. Again, this is a unilaterally treated eye, so you see the green arrows pointing to a relatively well-preserved outer nuclear layer over time versus the other side of the retina where we can see the thinning of the retina and especially the loss of the ONL in the untreated portion of the retina. This is a double injected eye and we see better preservation, and the course of disease is much slower when you inject and treat the whole retina. This is showing function. On the left, the visual acuity is relatively well preserved in the double injected mice, and also the ERG responses. And again, histology is perhaps the most convincing. So here's a unilaterally treated eye. So on the left, you see the treated side, and the photoreceptor layer is relatively well preserved, as opposed to about one layer of cells on the untreated side within the same eye. And here, a double injected eye on the right with preservation of the ONL versus the fellow eye in the same mouse, which lost most of the photoreceptors. And on the left, a graph showing quantification in a number of mice that carries through this message of preservation of the photoreceptors following gene therapy. And by using immunohistochemistry, we could show that indeed the Pha161A is expressed in the treated areas of the retina in the proper location in the inner and outer segments. And this also co-expresses with important other proteins in photoreceptors such as CRX, recoverin, and the different opsins. So it allows localization into the proper area in the photoreceptor and also function. So the mouse model shows that we can achieve slowing down of the disease by doing gene augmentation therapy. And we're hoping that we can now move to the translational phase in this roadmap and hopefully ultimately bring this treatment to the clinic. And again, thanks to the support of the FFB, we now have many, many patients that are diagnosed and could benefit for such a treatment. And to finish off, I would like to return to the slide that I showed earlier to give a little bit of hope here. I really think that you know, the treatments that we're looking for are not fast enough in coming. We all wish they would be here now or even yesterday, but I have a feeling that things are accelerating, and the FFB really has a major role over decades in supporting these efforts. And I thank you for your continuing support in this project and in others. And I want to acknowledge the many people who have been part of these efforts that I described here. My team, the CRMD, Dro's team, our department, which is very supportive, our Swiss collaborators, and especially the members of the IIRDC, the Israeli consortium, trying to pull together and identify and reach mapping of IRD patients in our population. Thank you very much for your attention. And now I'd like to move over and we'll ask Jason Menzo, who's the CEO of the FFB and driving all these wonderful projects we heard about, to replace me and speak to us. Thank you.
Thank you, Dr. Banin, and really thank you to all of our research colleagues from all over the globe. I certainly can't replace you. That is an absolute guarantee. That is not possible, but very pleased to be partners with you and really to all the researchers on today's call. As Dr. Banin had mentioned, my name is Jason Menzo. I am the CEO here at the Foundation Fighting Blindness, and I really do appreciate everyone across the globe for joining us today. As Laura mentioned at the outset of this call, we have over 400 people on today's call representing 45 different countries. Today's call will be recorded. We will put it up on our website. We will share. And, you know, one of the things that's really clear to us at the Foundation is, even though we're a US-based organization, that really inherited retinal diseases do not care where you're based. They know no boundaries. And our mission is to drive the research to provide for preventions, treatments, and cures, which is why we view it as such an important thing for the entire global community to come together on days like today to celebrate the progress we've made, but also to look at the advancements yet to happen. So we are going to shift now to the Q&A session. And I want to start by inviting all of our presenters to come off mute and turn their cameras on. We've got about 15 minutes for Q&A. And many of you have already chatted questions in, which is terrific. I'm going to ask Amanda in a second to repeat the instructions on how to ask your question. But one thing I want to ensure you is that if we don't get to your specific question on the call today, which we may not because there's a bunch of questions and we don't have limitless time, but if we don't get to your questions, we will follow up with you individually on email. And if you ever have a question, either today or in the future, you can always contact the Foundation Fighting Blindness, no matter where in the world you're located, at the email address, info@fightingblindness.org. So with that, Amanda, can you please remind everyone how to ask their questions, and then we'll jump right into the Q&A.
Yeah, of course. Thanks, Jason. You can send in your questions at the bottom of the Zoom window. There is the Q&A feature, and there's also a chat feature. You can use either one of those. And as Jason said, please send in your names and location too. We'd love to see where you're writing in from. And also info@fightingblindness.org. And we will follow up in the coming days or week.
Thanks, Amanda. Well, let's start. I'm going to actually direct the first question to our Chief Scientific Officer, Dr. Claire Gelfman. Claire, can you speak a little bit about the advancements that several folks chatted in, specifically around Usher syndrome type 2A? And maybe you can segue from the discussion around USH2A specifically into the gene agnostic strategy with oral NACA and the NACA trials that are happening. So maybe you could speak a little bit about that.
Sure. Thank you, Jason. Again, this is Claire Gelfman. So interestingly, one of our speakers today spoke about the fact that no matter what your genetic cause is for a diagnosis of an inherited retinal disease, a dying photoreceptor is what really causes a person to lose their vision. And no surprise then that a lot of the research has been around identifying the cause of that degeneration. And one of the implicators of that degeneration is something called oxidative stress. So no surprise then that there are a lot of approaches to treat inherited retinal diseases with an antioxidant. Now, I bring that into our discussion around Usher syndrome type 2A because there is a clinical trial right now in Australia, a phase 1/2 clinical trial that is evaluating an antioxidant in individuals with Usher syndrome, and the majority of patients in that trial have Usher syndrome type 2A. Now, this approach, though, is not only unique to Usher 2A. It's really for all types of RP, but there is a focused patient population in the clinical trial with those with Usher 2A. And this antioxidant, as Jason mentioned, is called NACA, N-A-C-A. The NAC part stands for N-acetylcysteine, and that's the antioxidant. And they made a slight tweak to that chemical that allows one to take it orally. So this is not an injection into the eye like a lot of the other approaches we hear about. It's an oral, you can take it by pill, and if approved, it will be one that can be given systemically in order to hopefully treat the retinitis pigmentosa component of those not only with Usher syndrome type 2A, but other types of Ushers, other types of RP as well. And this really stemmed from some initial data that came out of Johns Hopkins University, where just the NAC portion, the N-acetylcysteine antioxidant, was evaluated in a clinical trial that's now in phase 3 for retinitis pigmentosa that will also be applicable to all Usher syndrome affected individuals. Again, this is in phase 3. The trial is called NAC-ATTACK, and that will allow one to be given an oral pill for this type of RP. I wanted to also mention that through a big effort through the foundation, natural history studies can be used to learn about the progression of a disease, as well as ways to thinking about defining results for a clinical trial. And the foundation has sponsored one of these natural history studies specific for individuals learning of the progression of their Usher 2A. And we're now compiling all of that data so we can learn about the disease progression and then communicate that information with the FDA so that when clinical trials are posed, we can make sure that the right endpoints are included in those trials to identify success in these trials in a very meaningful way.
That's great. Thank you so much, Claire. Let's shift to a question for the whole group. A couple of different folks chatted this in, and it's interesting because it's become, at least here in the United States, and I'm sure it's the same globally, a very hot topic, which is artificial intelligence and quantum computing. And really the question is how you might envision artificial intelligence playing a role in advancing potential quick treatments and cures, maybe in your lab or in the clinical trial selection, perhaps in identifying unknown or variants of unknown significance or non-pathogenic variants that haven't been identified yet. But maybe a question for the group, just are you currently contemplating how artificial intelligence might play a role in your lab, or what thoughts you might have?
I'll start, Jason.
Thanks, Alison. I was going to say, I was waiting to see who was going to come off mute first.
So I think there's no doubt artificial intelligence has a huge role to play in aiding clinical diagnosis and outreach to centres that perhaps don't have the centres of excellence and the equipment that some of the big hospitals have. So this is crucial. In terms of the laboratory science, I mean, understanding the genetics, for sure. I mean, what we want to do is use machine learning, artificial intelligence to link the clinical phenotype with the genetics. So we have much deeper understanding, not only of that condition, but also the window of opportunity we might have, as we've just heard described by Claire, for the point of intervention. When is it best to intervene in that particular condition?
Great. Any other thoughts from our other presenters today on artificial intelligence? I think it's a hot topic, and it's one that
from our, I'm sorry, from our side, it's one of the caveats of all of these trials is finding a unique and a meaningful outcome measure. So right now we are trying to use deep learning and machine learning from OCT imaging to better understand how the retina remodels over time and can depict meaningful change that can be meaningful to detect disease progression and potentially drug efficacy.
That's great, thank you. Fernanda, actually, let me stay with you because a couple of questions had come in around the Stargardt clinical landscape, and in particular for younger children, maybe under age 10, for example. Maybe you could speak a little bit about that.
Well, being the ABCA4 one of the most common cause of macular dystrophies, there are many potential therapies, including gene therapy, stem cells, and pharmacological interventions. So far, most of them are in phase 2. We have one reaching phase 3, which means that if it works, it will be available upon FDA approval. And we have sense restorative therapies to disease, slowing disease progression as well. So the landscape is pretty bright. So far, we are still reaching adults and this trial phases. And I take this opportunity here to answer a few other questions that if you have a question from your specific gene, the Foundation Fighting Blindness website is an extremely reliable resource. We can go under the tab research pipeline, and you can see if your gene is there or not, or what a diagnostic therapies are. And so you can have very reliable and safe information to follow through.
Great, thank you so much. Dr. Hardcastle, let me come back to you, actually. You described in your work the idea of developing a retina in a dish. And I'm sure a lot of our audience is curious, how do you actually do that? It sounds like science fiction, but what is in lay terms, how do you actually accomplish this? And why does it matter for research? How can you use that tool?
Okay, so yes, it does sound like science fiction. And a few years ago, it certainly was. And that's why it's such an extraordinary advance in the field. Now, how do we do it? Well, we're really just following what we know about biology. We start as a bunch of cells and we end up with a brain and a liver and an eye and a retina. So, you know, how do these cells, how is fate determined in these cells? Well, we understand so much more about it now, that basically we can just feed these stem cells in a dish certain molecules, make sure we give them some nice food and give them some small molecules that essentially push them into a fate determination route that makes them into a retina. So we persuade them based on our knowledge of biology. Now, this does take quite a long time to grow a retina in a dish because we're recapitulating human development of a retina. So it can take many months, up to 8 months, to get a more mature organoid. But what it means is, for the first time, we've got photoreceptors in a dish, and we can make them, and we can make them again, and we can make them with different genetic conditions, and we can compare them to controls. We can understand how a change in a gene might be affecting photoreceptors, and then we can use these organoids as models to test therapies. And this has been proven that we can actually get proof of concept in a retinal organoid dish, retina in a dish model, and then translate that into a clinical trial.
Very helpful, thank you. Dr. Banin, you referenced in Israel, you've had great success at genotyping a large percentage of the inherited retinal disease population. Obviously, depending on the country that an individual is in, access to genetic tests may vary country by country. Here in the United States, we have a fantastic program sponsored by the foundation. Obviously, in Israel, you have your program. But the question is, when should an individual get retested? Because while genetics don't change, the technology to identify genes and mutations is rapidly changing. So from a practical perspective, when should an individual go back and retest if their initial test didn't come up with a pathogenic gene or a mutation?
Right. So indeed, over the 20 plus years that we've been working, the technologies have changed significantly, and we started with Sanger sequencing and homozygosity mapping, and over time, more and more opportunities. Now, exome sequencing and whole genome sequencing are potentially available. And the way we've been handling it is that we are trying our best to find the gene. If we're not able to find it in that certain individual, we usually take, a priori, we take a very large amount of blood and extract quite a lot of DNA. And as the technologies go forward, we revisit the samples that were not diagnosed and apply the new technologies, whether new sequencing or also new analysis that also comes along and is getting better over time. So I think this is something that, first of all, the labs or researchers that are searching the genes can take upon themselves. If it's up to the patient, it's sort of, you could follow a little bit the progress in the molecular genetics of these diseases. And then I think it's sort of reasonable every 3-4 years to get in touch with a physician and who's an expert in these diseases and consult with him or her whether it's time to maybe attempt again. But again, if the DNA is still in the lab that was working on it, revisit it. And we have families we recruited in 2000 we haven't solved yet, but many of these families from the early years were solved just in recent years as the technology's advanced.
That's great. Yeah, that's very practical advice. Thank you. Dr. Laster, I'm going to give you sort of the last word on questions today. Several questions today regarding XLRS and what's happening in clinical development and in the research pipeline related to X-linked retinoschisis.
Thank you. So very excitingly, a company, Axena, which is a gene therapy company for inherited retinal diseases, announced last month that they have dosed their first patient in a phase 1/2 gene augmentation trial for X-linked retinoschisis, or XLRS. And so just again, phase 1/2 trials, the primary endpoint there is safety. And then the secondary endpoint is to see if there is some visual function, if the treatment is efficacious. They are anticipating having 18 individuals in the clinical trial. So you can go to clinicaltrial.gov and search more about their study called the Lighthouse Study. You can enter in XLRS in the search field or Axena to pull up this particular trial. And that's true for all of the trials that you've heard about today, or as Fernanda said, even just your whatever your disease is, you can use clinicaltrial.gov to see what is upcoming, in addition to using the foundation's website.
Perfect.
Back over to you, Jason.
Thank you, Amy. So I know we have just a few short minutes left. I do want to introduce one of my good friends and colleagues, who is the CEO of Retina International, and that is Avril Daly. Many of you probably know Avril previous to becoming the CEO of Retina International. She was the CEO at Fighting Blindness Ireland. She runs a fantastic global organization at Retina International, which I get the pleasure of working closely with her in that capacity. And Avril, I'd love your thoughts on what you've heard today and really just your perspective on what's happening in the IRD field globally.
Well, thank you very much, Jason, and thank you to Foundation Fighting Blindness for putting on this website. It's this webinar. It's really encouraging listening to the researchers here today, and I think when we look to the community, we see, for example, a lot of hope around trials and treatments, and it's very exciting. But these are complex conditions. And I think what today demonstrates is we really need to continue our work and continue our efforts to understand what's happening at a very basic level and support basic research. And I think it's very exciting to hear what we have heard today. I really enjoyed the talks.
Sure. Amal, one quick question for you. So if an individual, because we do have many folks who chatted in saying, I'm in country X, how do I get in touch with a great local clinician or retinal specialist? Is it safe to say that individuals can contact, from the Retina International website to find the member organization that is in their region? So we have obviously Retina South Africa. We've got many organizations like the Foundation Fighting Blindness all over the world, and those colleagues of ours can help direct based on local resources that might exist in a given region. Is that fair to say?
Absolutely. I mean, Retina International has members on all continents, and ranging from very small voluntary organizations to very large organizations like yourselves in Foundation Fighting Blindness. And so we do get calls from people all over the world who are looking to find organizations to support them. And we put them in touch, and then those organizations will know who are the experts in their countries. But we also have countries that don't have organizations per se that we would be familiar with in European or contexts and in the United States, Australia, whatever. And they're really trying to find ways to develop. So it's important to be able to help them to develop organizations in order to be able to communicate to their community at a local level and engage their clinicians. So we're really building our community to be able to do that, because we get calls and we get emails, you can imagine, from all over the world, as I'm sure you do yourselves.
That's awesome. Thank you so much, Avril. And again, thank you to everyone for participating today from around the world. We, as I mentioned, have over 45 countries represented on the call. Really appreciative of our presenters as well for taking time away from their research to be here and share the great work that they're doing. And as I mentioned, while the Foundation Fighting Blindness is a US-based organization, IRDs do not care where you are based. And as an organization, we are leading the charge and really dedicated to driving the research to provide additional treatments and cures wherever in the world they can be found to advance the best science. So forums like the one we had today are a great opportunity to hear more about the efforts of the foundation and our partners as we continue to form alliances really all over the world. And we're not just focused on the clinical research for retinal diseases, but really bringing experts together to do so collaboratively. And as Laura mentioned, we're going to be kicking off some of our international efforts here at the foundation in the coming months, which will include some activities specifically in London. We've got a terrific kickoff event planned and more information to follow. You'll hear from my colleague in the months ahead, weeks ahead. Her name is Judy Taylor, who will start to promote and share with the community some of these activities. So thank you again for joining us today. I hope you have a great rest of the day, and happy World Retina Day. Thanks, everyone.